A common instrument in the biology lab is a spectrophotometer, often abbreviated as a SPEC. These instruments can be used to quantify solutions of cells, DNA, protein, pigments, the products of biochemical reactions, and more. To begin, the SPEC is turned on and allowed to self-calibrate. 15 to 20 minutes is often advised for the SPEC bulb to warm up to get a constant amount of light output. Before taking readings, the SPEC must have the proper wavelength selected and the absorbance set to 0 or blanked. A cuvette containing the negative control solution is placed into the SPEC, and the button labeled 0 absorbance, 100% transmittance is pressed. The blank cuvette can now be removed and replaced with the sample to be measured. Make sure the cuvette is clean and dry before taking a reading by wiping with a Kim wipe if necessary. Also, a square cuvette may have frosted sides, so make sure that the light path lines up with the unfrosted sides of the cuvette. Place the cuvette into the SPEC, close the lid, and record the absorbance reading. The SPEC can be used not only to measure constant absorbances, but it can also measure the color change during a chemical reaction. In this example, an enzyme is added to this cuvette to produce a yellow product. The reaction time would begin the second that the enzyme is added and may proceed for several minutes to monitor the reaction. After mixing the sample, place the cuvette into the SPEC and take readings over the course of the experiment. Here, you can see the increasing absorbance of the sample as the enzyme catalyzes the reaction. While the outer shape of these square cuvettes is the same, note that the first two have a V-shaped notch at the top to show which side should be lined up with the light path. The third cuvette is transparent on each side. Also note that the first cuvette has recessed sidewalls to allow a low-volume sample to achieve the necessary height to be measured adequately. In addition to square cuvettes, many spectrophotometers may also accommodate glass tubes for holding samples. After use, the sample may be rinsed down the sink if it is safe to do so. A bacterial sample would have to be disposed of into a disinfectant solution. These disposable cuvettes are all placed into disinfectant for disposal. Some common problems that may occur when using the SPEC are: if a sample is too concentrated, the absorbance may reach the maximum measurable value. For this SPEC, this results in the absorbance values flashing. The sample should be diluted and remeasured, as these values are not able to be measured properly in this machine. If the door of the SPEC is not closed properly, the machine will often show negative absorbance values as light is able to enter the machine. Note that this SPEC has the light path going from front to back, yet this cuvette is being placed with the sample window on the left and right. It will not yield the correct values for absorbance. If a cuvette is not seated properly in the SPEC, the sample may not be read properly, and sometimes not read at all. Always ensure the cuvette is pushed all the way to the bottom of the holder. Finally, if a cuvette does not contain enough sample, the light path will travel above the solution or hit the meniscus and give a false reading. Always use the recommended volume for the cuvette you are using.